Welcome to Beekeeping Today podcast presented by Bee Culture. Beekeeping Today podcast is your source for beekeeping news, information, and entertainment. I'm Jeff Ott.
And I'm Kim Flatham.
Hey, Jeff and Kim. Today's sponsor is Global Patties. They're a family-operated business that manufactures protein supplement patties for honeybees. It's a good time to think about honeybee nutrition. Feeding your hives protein supplement patties will ensure that they produce strong and healthy colonies by increasing brood production and overall honey flow. Now is a great time to consider what type of patty is right for your area and your honeybees. Global offers a variety of standard patties as well as custom patties to meet your needs. No matter where you are, Global is ready to serve you out of their manufacturing plants in Airdrie, Alberta, and in Butte, Montana, or from distribution depots across the continent. Visit them today at www.globalpatties.com.
Hey, thanks a lot, Global Patties, and thanks, Sherry. Hey, everybody, thanks for joining us. You know, each week we get to talk about how much we appreciate our sponsor support. They help make all of this happen and provide us the ability to bring you each episode. With that, thanks to Bee Culture Magazine for continuing their presenting sponsorship of this podcast. Bee Culture has been the magazine for American beekeeping since 1873. Subscribe to Bee Culture today, and while you're there, check out Bee Culture's Beekeeping, Your First 3 Years, a quarterly magazine for beginning beekeepers. We also want to thank 2 Million Blossoms to sponsor this episode. 2 Million Blossoms is a quarterly magazine dedicated to protecting all pollinators. Learn more in our Season 2, Episode 9 podcast with editor and our guest co-host, Kirsten Trainer, and from visiting www.2millionblossoms.com, and that is with a number 2. Hey, everybody, thanks again for joining us. We really appreciate you being here. We have an informative show all set for you with Dr. Boris Baer, University of California, Riverside, but more on that in a little bit. Hey, Kim, we're going into the final weeks of winter. How are your bees doing?
Don't ask, Jeff. I'm joining the ranks of those people on the 40% winter loss.
Yeah.
Very quickly. It's been a tough winter here. Cold, really cold. And I'll a side note on being really cold is my basement is really cold because it's not insulated, and because my basement's cold, my spring transplants are doing terrible.
Well, no.
But I think I've got it solved just in time. So I'll let you know how my tomatoes are doing in a couple of weeks.
All those heating pads underneath the flats are working, huh?
Yeah, they've been working, but the air's been so cold that it's been
not good.
Tough here.
So I was out in the bee yard yesterday, and you're talking about the 40% of dead outs. So what should beekeepers do with dead outs when they go out there and find a colony dead and moldy?
Yeah, that's it. So, there's a lot of things that people kind of don't think about when you're dealing with a dead out, and probably should. I am the world's, what's the term I want? I am the world's fussiest person when it comes to old wax on frames. Mm-hmm. 2 years and it's gone. I get rid of them. So there's all of that, and then there's the dead bees, and then what did they die of? And I'll tell you what, Jim, too, and I have been talking about this because we're both going through the same thing. And on our podcast, Honeybee Obscura, and coming up in a week or two, we're going to be talking in detail about how to deal with dead outs, because everybody's got them and everybody has to deal with them. So we've got some details that are worth listening to, I think.
That's really good because everyone experiences it. But I've not found much written about what to do with dead outs. They acknowledge them, but then they don't say what to do. So I'm looking forward to your podcast on dead outs. Yeah,
Jim had some good insights on some things. It was enlightening even for me, who's been dealing with dead outs for 40 years.
Very good. Well, I look forward to it. And speaking of current events, hitting the internet and different chat groups is a change in the Federal Registry of oxalic acid. Have you been keeping up on that in the last day or two?
Well, I've been trying to. There's a lot of information and a lot of misinformation out there.
Yeah.
I guess my only advice would be to make sure you know what you're doing if you're going to change what you've been doing with this stuff. I don't know if you can break the law or hurt your bees, so pay attention to the changes that are coming up this spring.
If you're into honey production, you're selling a food product and you don't want it to be tainted or get a bad name.
Or hurt yourself. That's
Yes.
that's the other thing that's going on. You can have a bad day in the bee yard with that stuff.
So everybody, you'll be hearing a lot of changes in the use of oxalic acid over the next couple days and next couple weeks. So make sure that everything's finalized and you understand and get your information from a reliable source before you change your management techniques. And finally, remember Brittany Goodrich was on our show earlier this year, this season, episode 19, and she was here on the show talking about the almond pollination contracts and economics of almond pollination. And she talked about a survey coming out this spring during to be timed with this pollination season in the almonds.
Did you hear what happened to her survey?
No. What happened?
Well, she put it out there, and I don't know enough about the world of malicious software, but something called bots got a hold of it and was filling it out and sending it back to her. And so the data was just made up. So it took her a while to figure that out. So then she redid it, and she gave it a link to go to the survey. And I guess, have you heard anything about? Did she talk to you about it?
Yes, she did send me a new link, and we'll have that in our show notes. So if you're involved in almond pollinations and as either a grower and a beekeeper or a beekeeper, she would appreciate for her research if you filled out her survey, and that link will be in our show notes.
We got a lot of good returns on the show we did with her. She's an economist looking at almond pollination, not a botanist and not a beekeeper and not a grower, but an economist. So the survey that she's doing, if you've taken a look at it, it's pretty intense. She should come up with some good information that'll help beekeepers and growers even more.
And while you're out there filling out Brittany's survey and checking out that link, also check out the rest of her website. We've got the new website up and going, and we'll be making improvements to that as we go along. And Kim, you mentioned Honeybee Obscure. How's that going?
It's going good. Jim and I are having a lot of fun, and we're covering some good ground. Like I said, we've got something coming up on dead outs, but we've done emergency feeding, and
good.
That's still an issue. In a lot of places, an issue where I am, there are people that are feeding right now because the bees are starting and there ain't much out there. It was 50 here yesterday. No, it was 60 here yesterday.
No way. Wow.
Way. And today it's going to be 21. So, it's all over the map and the bees are just going, what is going on? So if you go to Honeybee Obscura, we've got some timely topics in there that I think you'll find useful.
Yeah, it's very good. You guys are doing a great job. Well, Kim, you read more bee books than anybody I know. What have you been reading this week?
Well, I got a book just recently by a guy named Ed Simon. He writes a lot for Bee Culture magazine. And I got to tell you, Ed can make anything, and he can fix anything he makes. And I'm just in awe of the skill this guy has. Let me read you part of the review that's coming out in Bee Culture next month, and you can read the whole thing there. But if you got a chance before that, go looking for Ed's new book. It's called Build Your Own Beekeeping Equipment. And I think he published it himself because the only place you can get it right now is at www.lulu.com/shop. And we'll have that link on our webpage. You can chase it. It's 207 pages, $25. This is his second book on building beekeeping equipment, tools, devices, and things you never thought you would need to do, and some you'll be glad you learned how to build, use, and share. I don't think there's anything that Ed can't build, make, fix, or figure out. And this book is a compilation of his articles from several sources. Some of these are from his first book, Bee Equipment Essentials, published and still available from Wickwasp Press, and some are from his many contributions to Bee Culture magazine, and there's a whole slew of new ones. All told, Ed makes 10 pieces of hive equipment. That is, the boxes and all of those things. He makes 10 of those, starting at the bottom board and ending at the cover. When it comes to honey, he produces 8 different tools, including an escape board to get rid of bees out of supers, a drum dolly. He makes the most clever drum dolly you have ever seen. You got to take a look at it. He makes it out of stuff you can get, probably on the back of your garage. For wax, he makes a small-scale wax processor, a screener, and a strainer. Queens get some attention, too, with swarm boxes, mating nukes, even queen cages and sails. Sails, that's, how to build things for your farm market stand and in your store and that sort of thing. And he's even got a whirligig in there. I am a sucker for whirligigs, Jeff. I probably own 20 of them. And I cycle them all summer, and then you find out which are the good ones because you get a windy day and suddenly you've got 12 pieces of a whirligig spread out all over your yard. His won't do that. But I got to tell you, this book, as good as it is on the things that you're building, he's got the last chapter, and it's called Things Your Mentor Forgot to Tell You. And let me tell you about a million hints and tips on there that I'll just name a few. How to attach a wild comb to a frame, going from one hive to two or two hives to one, how to do that, and dealing with crystallized honey, and how do you clean your equipment, and placing hives in a bee yard, hive stand common sense, marking queens, making mini nukes, where to get free paint for your hives, dealing with pasture gates, hive cover. Well, it goes on and on. There's over 100 tips and tricks that your mentor forgot to tell you, and there's something in there for the biggest commercial beekeeper and the person who's still trying to think about maybe I should be a beekeeper. It covers it all. So, Build Beekeeping Equipment by Ed Simon, and you can get it on the webpage. You can get the link to where to get it on our webpage.
I know a lot of beekeepers like to build their equipment and have the ability, have the equipment. So this sounds like a perfect addition to their library.
Yep.
So, Kim, give us a little brief intro to our guest today, Dr. Boris Baer.
It's kind of out of the blue, out of the blue to me. The University of California's Office of the President scraped together $900,000 to develop new solutions for honeybee problems going on in California. It's involving 4 universities, and as I understand it, they're going to be, each university is coming at this issue of honeybee problems from a different angle.
Real good. Well, we'll look forward to Dr. Boris Baer coming right up. But first, a word from Strong Microbials.
Hello, beekeepers. Your honeybees face a lot of challenges out there. Unbalanced food sources from monoculture crops, holding yards, drought, food shortages, antibiotics, pesticides, and pathogens like chalk brood. To overcome these challenges, your bees need the multiple bacteria that are in all nectars, pollens, and the environment. These bacteria aid honeybees' digestion and improve your honeybees' response and resilience to pesticides. Now you can help improve your honey colony health with a quick, easy, and safe-to-use product. Strong Microbials Super DFM Honeybee uses naturally occurring bacteria to restore the healthy gut biome of your honeybees. Check them out today at www.strongmicrobials.com.
Hey, welcome back. We have with us Dr. Boris Baer from the University of California, Riverside. Boris, welcome to Beekeeping Today podcast. Thanks for joining us.
Well, thanks a lot for having me.
Boris, we've heard some news out of California that the University of California's Office of the President has given you a very large chunk of money to do some pretty sophisticated and complicated research with you and other researchers in California. I guess I'm just going to say, tell us what you're up to.
Thanks a lot. Yeah, I think we are very ambitious, but we got that money also because I think we have done a little bit our homework, and we can actually show that some of the things that we have in mind that is feasible and we can do that. And I think like we all start with things that we know that like we need bees for the production of our food. We like to keep bees, but these bees are in trouble. And I think we have a very good idea by now what the problems are with declining bee health and why we lose all these bees. This grant should hopefully, or not hopefully, is basically what I see like the turn of the tide in that like the research that has been conducted over the last couple of years allows us now to more think about how do we use that knowledge now to develop new tools for beekeepers. To monitor bee health and to maintain bee health, and also to kind of be better able to maximize bee health so that we don't lose these bees all the time, and that we have better ways to kind of keep our six-legged friends up there in the air. And this is what the grant is all about. So it's a collaboration between 4 campuses in California, UC Davis, UC Merced, UC Riverside, and UC San Diego. We have a team of very different people on there. We have like evolutionary biologists and ecologists, such as myself, but we have chemical ecologists. We have engineers on that team, and I think they bring all that expertise on the table to push this kind of tool development that we have in mind. And there are 16 projects in that grant. I don't think it makes much sense to go into each of these projects, but I think we have basically 3 main avenues that we kind of want to pursue. And that is, first of all, that we think there are bees out there surrounding us that are not managed, but that we know are actually quite good at defending themselves naturally against like environmental stressors. One example here in Southern California are certainly the Africanized bees or hybrids between Africanized bees and managed bees. We know that these bees must be able to deal with varroa. We know that these bees need to be able to cope with tremendous kind of heat here, exposure to pesticides. So why not climbing up there in the trees, get them down and basically ask them how they do that? And in the long term, basically try to use these traits for future bee breeding, so that we have like bees that can naturally resist or tolerate these environmental stressors. So that's one avenue that we want to pursue, which is more kind of what are breeding opportunities in the future for resilient bees. The second avenue that we want to take is based on research that we have now done for 10 years, not just in my lab, but also in other labs. And that is that we underestimate the honeybee. It has been around for 120 million years. It has seen a lot coming its way, and it always survived. And it is really successful, right? It has a global distribution at this point. So the idea that bees are completely defenseless is a bit naive. And so we looked at so what do bees produce when they get attacked by parasites, such as varroa, but we also looked specifically into nosema, and bees defend themselves. So we know that. Bees recognize these infections, they upregulate their immune system, and both for nosema and for varroa, we actually have identified the key ingredients that bees produce when they are confronted with these diseases. And in the case of nosema, for example, we know that there are molecules that the bees produce, and they are capable to kill 80-90% of nosema spores when they get in contact. So the second kind of route that we kind of pursue in this grant is to look for new treatment tools. Because when we know these bee-derived molecules, we can order some of them off the shelf. And that's what we have done already. And we can show that they would provide future new medications to manage bee diseases. And as a little kind of appendix there is we also are very interested in what is happening inside the gut of these bees, because we know this is kind of some of the initial kind of line of defense. These gut microbiomes, or these bacteria that live in the gut, they can provide some immunity, but they can also provide some protection, for example, against pesticide exposure and things like that. So there are quite interesting kind of opportunities to explore of how we could preventively kind of protect bees from environmental stressors such as parasites or pesticide exposure. That's a new tool new tools that we want to develop there to basically help bees in the future, based on the fact that for any other domesticated animal that we have around us, we have the veterinarian sciences, and they have a large amount of options of treatment and medication, but we don't have that for bees. And I think that we need to fill that gap. And then, so that's the second kind of cluster that we are after. And then we have the third cluster. And that's that I think there was the realization some years ago, actually, when we had these regular kind of meetings with beekeepers, that we are not really good at monitoring bee health over time. We all know that, right? It's like you have to open the hive. You might have to drive for kilometers or miles to actually get to them. And then very often you just stand there and you don't really know what's going on in a hive, or if your hive doesn't look happy, it's like, what exactly is the problem? So ideally, again, the veterinarians who kind of help us there, but that they have never really taken that up. So our idea was that we basically could shortcut that by developing sensors, electronic sensors that we place in the middle of the colony. And we kind of developed some sort of Google Translate for bees, right? So that we know that bees are very sophisticated in the way they communicate with each other, and they are able to kind of communicate about the status of the colony. We know that the larvae release a hunger signal. We know that larvae can release other signals that trigger hygienic behaviors when they're infected. We know there's a queen pheromone, right? So some of these molecules we already know. If we can build sensors that basically trace these molecules, the aim is to basically sniff into these hives and ideally recognize problems that occur in these hives as early as possible, and when we are still able to actually kind of counter upcoming problems. So these are the kind of three different kind of areas that we have been active in the past, and we leveraged the available information that we already have into the grant to now combine this all into kind of under the same umbrella. I'm super excited about that because it's it means all this work that we have put into this over the last years now really pays off, and we can really kind of start and get this over the finish line in the next 3 years where we get the funding for this.
Yeah.
Well, it sounds like, just roughly, it sounds to me like you're taking a lot of information, I'm not going to say unorganized, but isn't joined together well. The breeding problem or the breeding issues, that's been around, and people have looked at some of the aspects of Africanized bees and some of the feral bees, recognizing that they tend to do better than bees in boxes. And what you're going to look for is those things that allow them to do better than bees in boxes and transfer them to the bees that you're going to put in boxes.
Yeah.
Am I close there? Yeah.
Yeah.
And then the second sounds like you're going to harness the natural immunity that bees already have, and you're going to add to that some, I'm going to say chemical, but some level of protection against things that they can't control or can't. So you're going to give them that level of protection, and then finally you're going to take a lot of. I mean, there's a lot of people out there that are making sensors already. So you're going to take the best pieces of all of those and put them together and be able to manage not just this or that, but all of the things that are going on. Plus, it sounds like you're going down to the molecular level.
Yes.
And that I find exciting. If you can have bees telling you what's going on by the odor in the hive or the sound of the hive or all of those things, I think that would be, you put all those together and can you do this by June this year?
Probably not, but you're right? And I think that's the exciting part. And that's what I said in the beginning is the researchers, beekeepers have done their homework over the last 10 years, right? Since we know that we have these declines and we see these problems. So we don't need to kind of develop the wheel from scratch. I think it's a time frame now where we can put the technology and the knowledge that we have gathered over the past years together and actually make it work, right? And I think that was recognized in that grant application in that okay, you kind of promise us breeding programs and sensors and God knows what, right? It's it sounds a bit like Star Wars, right? And I think what we wrote in there is no, we can basically demonstrate that basic work has been done by some of it by us, but a lot of work by others, right? As you said, is others have started to think about sensors as well. So the idea is to consolidate that and bring the people together on one table and to basically get it get it done, right? And I think a very key ingredient that I haven't mentioned in the past is the collaboration with beekeepers. And we really push that so that we have constant feedback from beekeepers in the surrounding here, but also kind of from commercial beekeepers in that, like, whatever we kind of cook up here is not just kind of some insect or research nerds from a university, but that actually kind of has some link, or this is seen as like something that can be used and applied by the guardians of these animals, which are the beekeepers themselves. So we have built these kind of connections to the local beekeepers here. We have already run seminars with these beekeepers. We plan to do another kind of conference on bee health later this year, depending on how this all goes with the vaccination and with COVID. But I think we are very optimistic at the moment that I think by late summer, September or so, that we can see the end of the tunnel. We can actually sit together again in a room together without masks, hopefully, and
Yes.
And bring together, the students that we have here that work on these projects with the beekeepers and get that communication going.
Well, I hope by then you're able to pull that off and all of this craziness goes away. I'm going to go back to the breeding part of it, and I think that's where you're involved. In my experience over the years, that's always I'm not going to say most difficult. It's easy to breed a bee. It's difficult to breed enough bees.
Yeah.
And to take a breeding program and be able to get it distributed and make it consistent and then long term.
Mm-hmm.
And I've seen those come and go and come and go and come and go, unfortunately. Do you have some way you're looking at in terms of being able to be consistently produce bees that have the traits that you want?
Yeah, so first of all, there is a bit historic data for you. CIBER, that center that I lead here at the University of California, Riverside, has initially been initiated in Australia and was running there for, I think, 12 years or something like that. But it was the same philosophy that CIBER is the platform that combines beekeepers with researchers. And so our main link of CIBER in Australia was with the Australian queen breeding program, which is called Better Bees. And that's a queen breeding program that runs now for more than 30 years and has been extremely successful in that, like, they have wonderful bees. They produce hundreds of pounds of honey per year. They have hardly any losses there. Okay, they don't have a ROA, but for the environment where these bees live, this breeding program proved to be extremely successful.
I'm going to interrupt you for a second. How is that program managed in Australia? What's the background so that it remains consistent? Is it university or government or
both? And that's the exciting thing, right? So initially it was set up as an artificial insemination program to collect genotypes of interest, and that happened 30 years ago. And it was run by a governmental institution equivalent to the agricultural department. But it was gradually handed over to the beekeepers. So as of today, you have researchers and you have governmental people that provide input, but it's run by, I think, 8 or 9 different commercial beekeepers, and each of them maintain a certain number of lines. They evaluate them throughout the year for traits that they're interested in, or where we kind of provide them with some recommendations what they should select for. They then graft queens from the selected colonies and also kind of produce drone donors. And all of that stock is transported to an offshore island about 10, 15 miles offshore, where the bees are mated within the program. And that basically, so it's a closed breeding program that is run on an offshore island, and it has been extremely successful, as I said, over 30 years now, has really kind of pushed the quality of the stock up that is kept in Western Australia. So I think what I want to kind of say is we talked about breeding programs a lot. I think I've seen how such breeding programs can work. Again, one of the key ingredients is that it is unlikely that you run something like that at an academic institution. I think you have to involve beekeepers that are interested in that into it, and that you gradually also hand this over to beekeepers that are interested in maintaining such stock and developing it, but that you have like this constant kind of input from the research side. To make sure, for example, you're not running out of genetics, that you maintain sufficient genetic diversity. And with the tools that we have today, and that's some of the things that we have developed over past years to identify genes or genotypes of interest, right, that show specific kind of traits such as tolerance against, in this case, nosema, for example. I think this collaboration I think I see as like a model case that it is possible to run these breeding programs also over longer periods of time. And because the bees have these traits, these beekeepers can sell some of their stock off, or some of the queens, right? And they can generate some income through that, so that costs that are generated in that more economic kind of context that are actually met by some income.
Yeah, So you've got government and university backing, maintaining the quality, and you've got the commercial industry managing the quantity and continuously monitoring the quality. And the commercial group is able to make money doing that, and the queens that they sell, even those people are able to make money doing that, plus having a better queen in their boxes. How is the university and government program funded? Is that coming from directly from the industry, or?
Initially, in Australia, that was governmental funding. As I said, that's a couple of decades ago, right? Interestingly, when we were part of that, or when we worked with that breeding program, which is called Better Bees of Western Australia, the government had very interesting setups in that, like when beekeepers provided some funding, there could be either direct funding in terms of cash, or like they allowed us to access the genetics of their bees or some bee material for our studies. We could leverage these direct or indirect kind of contributions with the government. So for every dollar that we got from the beekeepers, I think we could leverage another $4 on top of this to do the actual research, right? So it was a bit of a tit for tat kind of approach. And so we got that type of money repeatedly. And that was really helpful, right? Because some of these analyses are very expensive, right? When you look at genetic kind of marker development, or kind of these molecular techniques, they can be very costly. You need to employ people. So I think that's there are kind of pathways how to finance that. And I think there are these opportunities in the US as well, right? You have Project APIS-M, you have NIFA, you have in California, you have more local kind of funding bodies that they can provide funding into these type of things. And typically, the way we operate, and that's the same for this grant here, is like you not necessarily get the entire amount of money you need from one source, but you basically kind of you get that together for a single component out of it. So if you look at the MRPI, for example, that funding that we got, that's substantial amount of funding, right? But it allows us to run the overall kind of umbrella kind of activities that we propose here. Underneath that, we have already applied for kind of segments out of that where we even received more funding than this initial amount to basically, fill the gaps and make sure that we have a continuation of that type of research.
Yeah, I can see how that would work. I'm hoping that you can make that work here. The other two aspects, essentially I guess for lack of a better term, call it the medications that you're developing to combat some of these problems. I can see where additional industries might get interested in producing those, chemical industries or drug industries. That would take that funding off off the backs of the beekeepers having to come up with that, and the government wouldn't be involved. And certainly the hardware that you're developing, again, it's the same thing. There are industrial organizations that would be interested in doing that. So if you can get the quality maintained by government and university and the quantity maintained by the commercial industry, working hand in hand, I can see where this could be very successful. I would like to see other problems in the bee industry attacked the same way, using the same techniques and management. I'm impressed. I hope it works for you.
Well, me too. But we set up CIBER here 3 years ago. Basically talked a lot to the beekeepers, adjusted these kind of concepts that we developed earlier in Australia to the local kind of conditions here. Clearly, Varroa, for example, was not on our agenda in Australia because Australia doesn't have the Varroa mite. That is now a big part of it, right? And I think the university has two advantages, right? The first one is because we are not an economic entity, we don't have to be profitable in a specific time,
yes,
right? So
exactly,
we have the breadth to basically really take the time to develop new sensors to search for these individual kind of molecules that bees use to communicate with each other. And we have access to bright people, right? We have students, we have PhD students that we can recruit into this that are highly motivated. The universities have these kind of offer these opportunities that you would not find in companies, either kind of chemical companies or companies producing sensors, or even in commercial beekeepers, right? We can offer that longevity, or we can generate kind of funding to get these things done through other sources, because that's in the end what we do. You mentioned the new medication, right? And I think that is also something that we followed advice from the beekeepers, which said, try to avoid to throw something additional in beehives that is artificial, and that we will have huge problems to get regulated, or it might end up in honey. Could you not find something that is naturally occurring in bees, right? So that was one of the things. And the other part is, of course, with these medications, beekeepers complain that medications or treatments that we use, for example, against Varroa, is starting to lose its grip. So if we have something that bees produce naturally, and that bees that we don't manage, maintain, if we see that some of these medications start to lose their effectiveness, we can just climb back into the trees and basically kind of see whether the non-managed bees have come up with something that has maintained its efficiency. So it's not like a one thing. We can actually kind of repeat that sneaking or spying on bee populations that survive without human intervention, and make that a continuous kind of process.
And you've got the engine driving this, of course, are people like you bringing graduate students in to solve those problems. They gain, you gain, the beekeeping industry gains. It sounds like a win-win-win. And for all three of those aspects of attacking this, like I said, can you have this done by June? I'll settle for July.
I doubt that we have it done by July, but I think, as I say, it's I see that this as the turn of the tide. I see that we have really wonderful opportunities now based on what we know about the problems, and the type of kind of knowledge that we have gained of positive kind of solutions against that. From my end, it's very exciting, because if you recruit PhDs at the university and they basically see that they're not just working on another academic problem, but there are beekeepers out there, and there is willingness to collaborate, and there is excitement about bees and about bee health. And there is this broader perspective, of course, that if you really find these solutions that will have a major impact on food security, on food prices, it can generate new jobs, right? So I think that what we get through that is also students that are extremely motivated, and that is then a real pleasure to kind of work with this type of students.
Are there time boundaries on the grants?
Yes. So this like initial amount of funding that we received, this will run now for the next 3 years. So it will make sure that we can really properly set these collaborations up between the 4 campuses, but also with the industry. As I said before, this is more or less the start of that activity. We're not stopping to apply for grants, and if we see opportunities, or if we see that certain things kind of start to fall into place, right, we're going to kind of follow up with additional funding applications to make sure that it stays alive. So for me, this is the start. I don't see this to finish in 3 years. We will do everything on our end to keep it alive, and a grant kind of success of that type is always good leverage, right? Other funders might see that and might recognize that, okay, somebody basically saw the benefits of that, and might more carefully look at what they fund or what they might refuse to fund.
So you mentioned the smells and the chemicals within the hive. Is that the only area you're looking at from a sensor standpoint? And sound, maybe?
I think where we see a real kind of need for development is sensor-based sensors that go for volatiles. And that's a bit more tricky because you need either to build sensors from scratch that go, for example, for the queen pheromone or for a hunger signal or things like that. So you either build new sensors or you retrain sensors that are already out there. But we also we played around already in the past. I think over the last 5-6 years, we have just deployed sensors that you can buy and that you can deploy, right? To see where do we place these sensors? How long do we have to take these data? And you have to see that the data collection per se is not the end of it, because the beekeeper or the person who deploys a sensor like that doesn't kind of want to sit on the screen at home and then start to do statistics on this to figure out what the heck's happening.
And you're right there.
Yeah, exactly, right. So, and the other thing is if you sit on your desk and you look at what's happening in your hives, and in the evening the software tells you your colony died today, is that's not good either, right? It's yeah, that's nice, but it would have been nice to actually know that much earlier on. So I think once you collect the sensor data, and that's what we have done now quite a lot, is we use this data and we hand this over to engineers that are familiar with artificial intelligence and machine learning, right? So they kind of use these approaches to basically do a software that is doing 2 things. First of all, it combines all the sensor data that you have to make an accurate kind of prediction of what is happening. And that might not necessarily be driven by a single sensor. If you're sick and you're going to your doctor, he takes your temperature. But just because you have a fever doesn't mean that the doctor will exactly tell you, you have a sniffle or you have cancer, right? So he will make additional kind of measurements. And depending on what he gets back, there will be a picture is emerging of what the problem is. It's a bit the same with bees. So these artificial intelligence people, they need these large scale data sets, first of all, to make an analysis so that the beekeeper, for example, on his mobile phone would get an exact kind of statement of what's happening. Your bees are hungry, or you lost the queen, or your varroa levels are too high, right? Things like that.
You want it to be predictive.
Exactly. And
to give you a warning before it happens.
Absolutely. That's the second thing. And that's where we've really pushed the engineers by saying, it's not enough that you tell us now that the varroa levels are too high. We need to know this well in advance. We would need that to a level where the colonies are not collapsing or have not, lost the queen. So these people basically at this point, they just want to have large data sets measured for many colonies over prolonged periods of time, which we have, so that they can start to play around of how much variation do you have inside the colony? How much variation do you have between colonies, right? There is the approach is to most likely have multiple sensors in an apiary, and then you have some sort of a baseline, and the software basically looks at, okay, which colony is basically starting to deviate from what's happening in, and then give an as early as possible kind of warning signal to beekeepers.
The next challenge after setting it up for the engineering is to bee-proof and beekeeper-proof any sensors in the hive.
Yeah. So our idea is that and that came from the beekeepers as well. It's like, if it's too complicated, forget it, right? We are not going to kind of have 15 miles of cables and antennas and things like that in our beehive. It would have to be, certainly in a commercial aspect, it would have to be affordable. And the beekeepers that we talked to actually made some back of the envelope calculations of what such a sensor should cost in order to offset other costs that they would have with replacing queens or sending people out. Right? I think we came up with something like a maximum of $60 per hive, which would be the threshold where it becomes actually cheaper. But the other thing is like, and that's, we have other types of engineers on there, which like where it's very clear it has to be fully integrated into the usual kind of beehive activities. So our sensor would sit in a length-trough frame in the middle of the hive. We can miniaturize it so that it's not taking up too much space. The energy would not come through a cable. We're playing around with things where we could kind of send electricity into the sensor wireless with some solar panels on the roof. Right? This is the type of challenges that I think like are there, and that's the way you get the engineers excited, right? Because it's like, because it causes challenges also for but that's what also the graduate students or the students in engineering want to do. They want to see some challenges.
Well, this is exciting, I think, Boris. I am like I said, I'm hopeful that it works, and you seem to have a lot of the I's dotted and the T's crossed already and ready to move forward. I don't want to take more of your time today, but we want to get back to you in a while and see how this is going. And July, being done by July is soon enough for me. That would be good.
Okay. We try our best, but just keep in mind that we are still in lockdown. Is like we eventually need to be able to send students back to bees to be able to be back in the bee yard. But even though we sit at home, for example, working on these sensor data files that we accumulate, that is still happening. Right? So,
well, definitely a lot of work to do yet. But I see the three different areas you're working on, and then ultimately the goal would be to have them all sharing information across the different bee types or the natural bees, so the integration with the Africanized honeybee and the natural defenses and tying that into any kind of sensors and have them all read and working together. It's quite ambitious, but it'll be fun to see the progress.
I agree. Yeah. So I think like throughout my career, I started my PhD when these first kind of reports came in about declines in honeybees and later on also in native bees, and I felt it to be very frustrating. And you know, you were working in that area, but you basically were just kind of managing death around you. And I think that has changed over the last few years. I really think like we know the issues, and I think we can turn the tide. And I think that makes a huge difference.
That's a good phrase, turning the tide. I like that.
Yeah, it's a nice positive message.
Yes.
Yeah, thank you. Well, we definitely appreciate you being here this afternoon. Look forward to having you back on the podcast and hearing the developments and the findings of your continued research. Thanks for joining us.
We'll be in touch.
No problem. Thanks. Bye-bye.
Well, I couldn't think of a better person to lead up that team than Dr. Boris Baer. It sounds like he's got a great plan in place.
Well, the plan, I'm impressed with the plan. They're coming at it from breeding bees that are resistant to the issues that bees are having, primarily Varroa, bringing in some of the, how do you say this carefully, the good points from Africanized bees that seem to show some resistance. Once they get that, they're beginning to look at new or improved medications to treat the issues that bees are having brought on by Varroa and other things. But so there's some breeding and there's some new medications, and then they brought in a whole bunch of engineers, and they're looking at the electronics of monitoring hives. There's a lot of stuff out there now already, and I wonder how much of that they'll be able to improve as opposed to just being able to incorporate maybe 2 or 3 or 4 or 9 different techniques to find out what's going on in a hive. It's pretty impressive. If they all work, I think we'll be better off when it's done.
Yeah, well, it's definitely, as we've learned over the last year and last couple years, seasons of the podcast, the research that's going on right now in the bee field and for pollinators is pretty exciting. It's probably never been greater than it is right now.
Yeah, it's I don't know what these engineers are going to be looking at in California, and we're going to go back and talk to Professor Baer here in, I'm going to say, a year or so to see how they're progressing. I think it's a 4-year grant that they've got. Don't hold me to that, though. I'm not sure I remember that right, but it's several years.
Well, that about wraps it up for this episode. Before we go, I want to encourage our listeners to rate us 5 stars on Apple Podcasts wherever you download and stream the show. Your vote helps other beekeepers find us quicker. Even better, write a review and let other beekeepers looking for a new podcast know what you like. You can get there directly from our website by clicking on reviews along the top of any web page. As always, we thank Bee Culture, the magazine for American beekeeping, for their continued support of Beekeeping Today podcast. We want to thank our regular episode sponsor, Global Patties. Check them out at www.globalpatties.com. We want to thank Strong Microbials for their support of this podcast. Check out their probiotic line at www.strongmicrobials.com. And finally, we want to thank you, the Beekeeping Today podcast listener, for joining us on this show. Feel free to send us questions and comments at questions@beekeepingtodaypodcast.com. We'd love to hear from you. Anything else, Kim?
Yeah, I think that about wraps it up, Jeff. I'm going to try and stay warm the next few days.
Yeah, go down there and warm up the air around your plants. All right, take care.
You too.